So hello students and welcome to the Next Generation Sequencing course week 11. Today we'll be talking about the statistical aspects of RNA-seq data analysis. I'd like to just begin with an overview to add some continuity here to some of the topics that we've been talking about. So what is the point of these RNA-seq analyses, and how do we execute an RNA-seq analysis? Well, generally the point is to gain some biological insight into gene expression differences between samples, typically in a treatment and control condition. And we begin by collecting RNA from multiple samples of biological replicates in each condition, and then we extract RNA. We go through the process of building the RNA-seq libraries. We conduct the sequencing on a short-read sequencer, and then we go through the steps that we've been spending a bit of time with here in this class, such as the alignment protocol, including bow tie or top hat in this case. Unmapped reads from the initial bow tie run in top hat are then mapped to the splice junctions. We then collect a table of counts, of read counts to each transcript, which is our measure of gene expression since RNA-seq. In principle, each read will represent the presence of a single RNA-seq molecule, RNA sequence, or molecule in the original cell. And then we take this table of read count data where we have read counts for each gene or each transcript in the cell, and we need to conduct statistical analysis to determine if genes are differentially expressed between different treatment conditions. And in that, if they are, we then conduct analyses to infer which biological pathways might be differentially regulated between two cells, or go on and infer regulatory networks using the RNA-seq data themselves. So that's just the overview. This is also a review slide to indicate why RNA-seq is a important method now in molecular biology. There's more reasons than this, but these are two reasons why people began to abandon microarrays. Was simply because of the high technical replicability of RNA-seq. So this is coming from two technical replicates of brain cells. Each point on the slide is the RPKM from technical 1 and RPKM of technical 2. Technical replicate 1 and 2, and so you can see the really strong correlation. The other reason, in addition to a few others, another reason anyways, is the dynamic range in RNA-seq analysis spans roughly 5 orders of magnitude. What this means is that we can still infer the difference in gene expression between samples, even at quite low abundance of transcripts. And we'll talk about some of the issues related to that later in this lecture. So one of the things we have not talked about is essentially how do you actually get the read counts in cases where you have complex mixtures of various transcripts. So we have talked a bit about alternative splicing, but I haven't told you how different people have accounted for alternative splicing in coming up with their read count estimates. So what I should point out is that the method that we've been working with in the homework, the isoform-specific deconvolution methods, is a class of methods of which cufflinks is a member. This is a fairly sophisticated methodology that has only recently been developed, okay, in the last year or two. Whereas before that, we had these methods where the problem was we had, if you have a complex mixture of isoforms and you want to come up with a single measure of gene expression for each gene, because we didn't have the capacity 3 or 4 years ago to infer individual transcripts and to model them, which ones are there, and to then count individual ones, which is what cufflinks does. People were tried to take some, rather simplified shortcuts to still, getting measures of gene expression for individual genes. And so let's take a look here at an example where we have isoform 1 and 2, so these are two alternative splice variants from the same gene. Notice that light blue represents an exon that's found in isoform 2, dark blue an exon that's represented in isoform 1. And so this is the way in which this gene can be alternatively spliced. And so how would researchers then go about counting reads to come up with a measure of gene expression for a particular sample? Well, or particular gene, rather. Here you have the exon union method, where the researcher would simply take all exons. All 5 of them are here. It doesn't matter which isoform they're in, and just tabulate the total number of reads. Another method would be what's called the exon intersection method, where a researcher would simply take what we call the constitutive exons. Those are the exons that are found in each transcript, and would count only the reads from those constitutive exons. Now, cufflinks, quite carefully in the original cufflinks paper, which I am inclined to post now. I have not posted it as of yet, but I'm inclined to post it simply because it's, I think, a significant paper in bioinformatics and gene expression analysis. But what cufflinks pointed out was that these simple methods of dealing with alternative splicing actually lead to misinference in many cases, and so they outline why that is, and then they motivate that motivated their much more sophisticated approach to modeling individual isoforms, and then using a statistical model to estimate the number of reads that are associated with each isoform, which of course is not a trivial problem, simply because when individual exons are shared and you don't necessarily know which transcript exon 1 actually came from isoform 1 or isoform 2, it becomes non-trivial to estimate the number of transcripts, or reads associated with each transcript. But in any event, that is what cufflinks does. So anyway, hopefully that provides some historical background on the problem, and how now methods like cufflinks have quite sophisticated ways of obtaining read counts for individual isoforms, and of course, as you know from the cufflinks outputs, that you actually get individual estimates not only per isoform, but also cufflinks will sum across isoforms and give you measures per gene based upon those modeled estimates from individual isoforms. So before diving into some of the statistics, I thought I'd make it clear on exactly the type of experimental design we're talking about, and what experimental design we're trying to obtain statistical significance of gene expression differences between treatments. So this is kind of a fairly standard, simple but standard experimental design that is currently being performed in RNA-seq analysis. There's certainly, all kinds of more complicated designs that might include multiple, experimental groups and multiple sort of hierarchical levels of organization, among samples. And yet here we simply, generally in RNA-seq, the state of the field at the moment is that the statistics and analysis packages are basically set up to conduct differential expression between a control group and an experimental group. So remember, in our rice example, we have a set of rice that are growing at one temperature, an experimental group in which a set of plants were transferred to a higher temperature. And so these are replicates, and the replicates are important. It's something I want to emphasize here. You cannot just take a single plant at one temperature and compare it to another plant at a second temperature, and then try to get differential expression across treatments in that statistical design. And the reason is because we can't distinguish differences that are simply due to biological replicates within a temperature from differences that are associated with the condition that we're actually interested in measuring. So we need multiple samples. So in our homework, you had 2 rice samples, which is enough, not ideal, but enough to estimate the variance within groups and between. Okay, so you had 2 control and 2 experimental group samples. So these are biological replicates. It's unusual at this point to include technical replicates, where you simply take RNA from the exact same plant, the exact same treatment, and then simply sequence it twice, or isolate RNA twice from it and conduct the entire sequencing protocol. That would be a technical replicate. We generally don't do that at this point because it's not necessary for the statistics, and it's just very expensive to do. So in any event, we have a 2-sample design. I thought I'd also point out with replication, with biological replication in each condition. And then I should point out that we have to take into account this important concept of randomization really throughout the entire preparation of the libraries and sequencing step. So a bad design would be for a technician to come in and on first day of the experiment prepare libraries for the control group, and then on day 2 prepare libraries for the experimental group, and then put the libraries from the control on one lane of the Illumina, for example, and then the experimental group on another lane. What that does is that introduces what we call in statistics block effects, which are highly undesirable, and it's important throughout the experimental design to remember that you want to avoid those block effects by randomization. So what that would lead you to, or at least make you susceptible to, would be to inferring differences that are between what you would infer to be the biological condition of interest, that is temperature, in our rice experiment, and it might lead you to infer that there are temperature differences when in fact those differences that you infer are simply due to the fact that samples from the experimental group were on one lane and samples from the other group were on another lane, and there may have been systematic artifactual biases that lead you to draw the wrong conclusion about where the source of difference between the samples comes from. So a good design is to simply randomize. So, and the hypothesis, remember, that we're interested in testing is, does a gene differ in expression in the 2 treatment conditions? And we're interested in evaluating that hypothesis literally over every expressed gene in the genome. So there's essentially 3 steps to conducting statistical tests in RNA-seq data. We have to normalize the read count data. We have to infer model parameters, where we need to estimate the variances in read counts across the samples. And then we have to conduct the statistical tests of significance. So we'll be going through these in some detail in the next 20 to 30 minutes or so. I'd like to begin just by illustrating the point of why we have to normalize the data. So there's a few really important sources of bias that would lead you to draw errant conclusions about the data if you didn't normalize. So in this hypothetical example, I'm showing you the effect of length bias, which we have to try to control or remove from the analysis in order to conduct the tests. So imagine gene A and gene B. They are simply of different lengths, although in this hypothetical example they are expressed at the exact same level. Imagine that. And in this case, simply because gene A is longer than gene B, more of its copies of gene A's transcript end up being represented in our transcript, in our RNA read count, simply because the gene is longer. Okay, so here we have 30 reads, 15 reads, which makes it look unnormalized anyways, that gene A is expressed at a higher level than gene B simply because it has more reads, right? So we simply have to remove this length bias from our analysis. And so this is generally what we call a within sample source of bias. That is, really notice that we're contrasting two genes here, not contrasting two samples, as I mentioned is our typical design, where we're trying to contrast across experimental conditions to see if there are differences in gene expression at one locus, not comparisons between two. But in any event, we do generally try to account for this obvious source of error in our normalization protocol. Although we generally expect that because gene A is what we're comparing between two samples, that the gene A is the same length in both samples, and therefore at least we anticipate that it should cancel out between sample comparisons. So how do we deal with the problem? Well, I have seen this done. This is certainly not the common way to do it. You could only analyze portions of genes such that all genes have a common length. So we could say take some fraction of this gene and take some fraction of gene B, and we always take the same total length of sequence from each gene to analyze across our entire data set. Of course, that seems kind of like a boneheaded approach, and in fact it probably is, because you're losing a lot of data from the remainder of the genes. So that's generally not how we do it, although it has been attempted. Another way to do it would be to scale by length, and this is what we generally do. So you simply will divide the number of read counts per gene by the gene's length. So another problem that requires normalization is that there are differences in library size, so the overall sequencing depth. And you might say, well, we can deal with that just by tossing out a subset of the reads from the deeper sequenced libraries such that all libraries have the same size. Well, again, that's a bit aggressive handling of the data, because you're losing a lot of information when you do that, and we generally think that more information should generally make our inferences better. So the approach is to try to normalize then for this library size effect. So let's see, why do we need to normalize for this library size effect? So first, imagine two samples, in which we've extracted RNA, done our sequencing, and we realize that sample B has just higher depth. We just had better cluster formation on the flow cell. We just got better coverage of the overall library. And so here you'll see that for each of the two hypothetical genes here, that sample B has twice the number of reads as sample A. But what I'm illustrating in the hypothetical example is that two genes here, one gene in red, one gene in blue, that are not differentially expressed. But just to make it clear, there are 60 reads here, there are 30 here. There are 30 reads in the blue gene B here, and 15 reads in the blue gene B in sample A. And the point is that without correcting for the library depth difference over the entire genome, we might be tempted to think that sample B is overexpressed for each of these two genes compared when we compare them. So this is a clear bias here that we have to take into account, and it is in fact this library size effect that is the primary source of problems in normalization, for reasons that I'll get into in a moment. So we have high read counts in sample B that are attributable to greater read depth across the entire library, not differences in gene expression. And these bias effects comparisons of expression levels between samples. So in contrast to the length effect, which I explained to you as being mostly an effect associated when you compare genes, or problematic when we compare expression levels within samples between genes, this is a problem that clearly affects our ability to contrast expression levels between samples, not within samples. So there's a problem, though, with total lane count. This is the term that's used for these RPKM, FPKM normalizations. That is, we're normalizing generally by the total count. So let me just, before we go on, let me just go back to the RPKM and FPKM ways of normalization. Reads per kilobase of exon per million reads. So per kilobase of exon, the way that we generally account for the length effect is to simply divide by the length of the gene. And the way that in the RPKM strategy we deal with the the problem of differences in total library coverage, we just divide by essentially by the numbers of reads. Okay, so divide by the length, divide by the number of reads, and then now you have your normalized measures. Well, that was used for several years until researchers began to think a little bit harder about the problem and realized that there is a major problem, and that is the biological observation that highly expressed genes really do make up a very large fraction of RNA-seq read pools. Okay, so in a study of the human brain, for example, 5% of genes make up, up 50% of the total reads from a human brain tissue. In our laboratory, we see the same effect, but for a gene involved in photosynthesis. This gene Rubisco is a multi-subunit enzyme that, essentially sequesters carbon dioxide and builds it into sugars, and that's the role of Rubisco. It's a very important gene, and it's highly expressed. It's massively expressed, probably the most expressed gene in a leaf tissue of a plant, anyway. And so what's the problem? Well, the problem is that read counts, essentially are not independent between genes. So read counts, being our measure of gene expression, are not independent between genes because there's a fixed number of reads in a library. Okay, so if we have a fixed number of reads and some huge fraction of those reads are going into sequencing Rubisco, and some smaller or lower expressed gene gets proportionally fewer reads, okay, then the two are not independent, because if the read count for Rubisco goes up, then the read count for the gene of interest, perhaps that lower expression, goes down. So there's just fewer reads that are available. So the problem, really, the proximal then problem is that differential expression, of the very highly expressed genes is the problem. Okay, because if you have a gene that's very high transcript abundance and is represented in our libraries by a lot of, or a huge fraction of the reads, and one sample has higher Rubisco expression biologically than another sample, essentially you can think of the problem as, if we're just basically normalizing on Rubisco, because Rubisco makes up such a huge fraction of the library. If we simply divide here by the the numbers of reads, effectively we're just dividing mostly by Rubisco's read counts. And so if Rubisco is differentially expressed, then we are effectively you are not normalizing the same between each of the two libraries. Because for the sample that has higher expressed Rubisco, we're normalizing by a greater extent, or we're dividing by a bigger number than in samples that have lower expression of Rubisco. And so this is the problem, and it's really the basis for development of a number of alternative normalization strategies. So we still see RPKM and FPKM, right? We see these in all the papers, but the thing I just want to emphasize here is that these are really, what we might call effective RPKM and FPKM, and they are adjusted. Typically in studies that you read now, these are not the raw RPKM, FPKM values. They are scaled by some factor, but we still keep the terminology RPKM or FPKM, but they are adjusted values, so we might better refer to them as, say, effective RPKM or FPKM. So here's how, I just thought I'd give you an example. Some of this gets a bit complicated, and I probably will try to avoid explaining all of this in words or in any sort of detail, because it's something that you are better probably just spending some time with thinking about, particularly in the package that you're interested in using. But I just thought I'd give you an example of how we might go about adjusting the FPKM or RPKM by scaling factors. And just to take an example, DE, or differential expression seq, is an R package. It's one of the 3 or 4 most commonly used ones. It's obviously separate from the cufflinks, cuff diff approach that we're using in the homeworks. But the DE-seq does have a scaling factor, and the scaling factor is calculated on a per sample basis. And so, if I draw your attention to the red line, what DE-seq does is on a per gene within this sample we're talking about, it takes the read count of the gene. It then divides the read count from that sample by the geometric mean across samples for that same gene. Okay, so this is a per gene ratio here of read counts divided by the geometric mean across samples, and then the scaling factor that's applied to every gene within that sample, okay, is the median of the value of this ratio, which is calculated for each gene, okay, within that sample. Okay, so and then that's the scaling factor that's simply applied to the FPKM and RPKM to get the values that might be reported by the DE-seq package. So Cufflinks actually uses and CuffDef uses a similar methodology. I believe it's slightly more complicated than the DE-seq approach because it sort of scales, how this geometric mean, I believe, comes from different samples, and then there's actually two scaling factors. But this is a simpler example of how these scaling factors get calculated. Okay, there's other methods here like quantile normalization. This is a method that was commonly applied in microarray data analysis. Upper quantile normalization, there's a few of the papers that I posted online actually go into some of these other methods in some detail, and of course different authors make the case for different forms of normalization, and yet each of them is subject to their own biases and their own problems and are probably less than perfect. But this is sort of the state of the field at the moment in any event. So another approach, I just thought I'd contrast this with how things might be done, in other experimental protocols, but are typically not done in RNA-seq, is to normalize to housekeeping genes. So what is a housekeeping gene? A housekeeping gene is a gene that we generally assume is expressed the same in each of our different replicate samples, including across our treatment conditions. And if we can see that one of these genes changes in gene expression across samples, then we simply account for that as a technical problem that needs to be factored out when we analyze other genes, okay, because we assume the genes are in fact equivalently expressed across replicates and conditions. And so we can use that difference in expression of these housekeeping genes then to develop scaling factors that we can use to normalize the data based on the housekeeping genes. This is something that we do more commonly in quantitative PCR, but is not generally applied in RNA-seq data analysis. So I just thought that this was a helpful figure to sort of shed some light on the normalization process and what you get in the outputs and how the outputs are different from the inputs. So here's the raw counts, unnormalized for length or unnormalized for the size of the libraries. Here's the RPKMs. I should point out, each of these samples comes from a mouse. I believe it's a brain. And each sample here represents the distribution. It's a box plot of read counts. So the upper part of, you're looking at the 75th quartile, the 25th, and here in the medium is the median. Here is the black line. And so you can see that these are unnormalized samples here. The distributions of read counts are very different. And for reasons I've already mentioned, you can't then take data like this and try to make biological inferences because you end up with a lot of artifacts due to library size problems, gene length problems in the raw counts, etc. Okay, so we have to normalize, and here after normalization, these are a number of the different normalization strategies that people have applied. And so here, this total count, I believe, is just when you normalize by library size, but you don't normalize by gene length. Anyways, you can see that for most of the normalization protocols here that are commonly adopted, these ones here, that you do at least get rid of some of the major problems that are associated with the raw RPKM and raw counts. So this is one of the papers I post online, just how to take a peek at it if you're interested in more detail. So so that's the normalization step. The second step that I mentioned is that we need to estimate parameters from the statistical models. So this is primarily the variance. The mean and variance are the parameters of the models that we're interested in. So I should point out that count data do not fit continuous distribution, so we generally don't use the normal distribution in our analysis. And instead, researchers initially began to use the Poisson because it was thought that the Poisson would account for the variability that we observed in RNA-seq data. And it does mostly account for variation across technical replicates and within samples across genes, but the variation in read counts that we see across biological samples and across treatment conditions is what we call over-dispersed. So just for review, or for those of you who are new to statistical modeling or new to statistical distributions generally, keep in mind that some of this is technical detail that I'm presenting for sake of completeness, and not something that as bioinformaticians you necessarily need to know, but I think understanding some of these basics is important. So the variance in the Poisson distribution is equal to the mean. And so when we find cases where the variance in read counts across genes or across samples is greater than the mean, okay, then that's evidence that our data set is not, conforming to expectations of the Poisson distribution. And so that has led to a general abandoning of the Poisson distribution in terms of modeling RNA-seq data, and instead researchers now are widely adopting the negative binomial. So the negative binomial has variance equal to the mean, but it's scaled here by the mean squared and this what's called dispersion factor alpha. So this dispersion factor alpha really is the thing that differs between the different packages. So all of the different statistical methods now, or the majority of them in any event, use this negative binomial, and they differ really in how they go about estimating this factor alpha. So then once we have the model and we have estimates of the ways of estimating the variance in the model, we then can test for differential expression of genes in our experimental design that I showed you, where we have treatment versus control. Different softwares use different statistical tests. Some use Fisher-Exact, some use likelihood ratio testing. Cufflinks, I just thought I'd give you the example since it's the one we use in the class, uses a t-test. Now, t-test assumes a normal distribution. What's normally distributed in this context is this ratio. So E here means estimate, and the Y here is the ratio of normalized counts between the two conditions. Okay, so for a gene, we have counts for a gene in condition 1 and counts for a gene in condition 2 across replicates. We then have the variance estimates for this ratio, and from that we can come up with this t-test statistic. The statistic is normally distributed, so we can use the same distribution that we would use in the context of a t-test. And so this is how we go about assessing statistical significance in cufflinks. So of course, when you look at the raw output data, cufflinks has done all of this for you. You don't ever need to type in or to evaluate this yourselves, but you simply get a p-value out. But this is sort of where the p-value is coming from. Okay, so without going into or trying to explain the extreme details, these are things that perhaps we can talk about in more detail if you're interested in it. But these are things that are also generally not absolutely mandatory, because we, as bioinformaticians, when we're trying to execute and learn how to execute all of the analyses, sometimes we are forced, really, to trust that the statisticians who have developed the software packages, have correctly, developed the statistic. And so, while it's always good to be able to think critically about how this was calculated, in practice, you'll find that many people in the bioinformatics field generally, take it for granted that these calculations have been done correctly. Okay, so I just wanted to introduce a few problems because, and I'll use one example here because I think it's essentially a way of illustrating that the RNA-seq statistical analysis field is very much in development and there are still problems. So one of the problems is this, as pointed out in this paper that I posted by Bullard et al., is that standard errors of our estimates, in read counts have an inherent length bias, where short genes have larger standard errors than longer genes, and as a consequence, longer genes are more likely to be inferred. It's not like they're more likely to be differentially expressed necessarily, although that's certainly possible as a biological factor, but we're talking about a technical artifact where longer genes are more likely to be inferred to be differentially expressed because of the smaller standard errors associated with these longer genes. So that poses a problem for interpreting gene lists when we go in and we have identified a gene list, a full set of genes that we've identified differ in our treatment conditions between conditions, and then we're trying to interpret that list as and identify biological pathways that tend to be differentially regulated in that experiment. Well, the problem is that what if the gene lists here tend to be sort of enriched for genes that are just longer because of statistical artifacts associated with this problem? Okay, so we'd like to be able to interpret this gene list strictly in terms of biology, but there are oftentimes issues that you encounter, such as this length bias effect that makes it, you at least have to be careful in over-interpreting gene list that you derive from this type of analysis. So I thought I'd introduce now, this is a topic that is not specific by any means to the RNA-seq analysis field, but one that is generally an issue in genomic research, certainly, and in many areas of statistics, where we are conducting many independent tests, effectively, it's a family of tests, you might say, across, in our case, the entire genome, where we're conducting a t-test in cufflinks or cuff diff, across every gene in the genome. And the problem with that is that, the null hypothesis of no difference in gene expression may be rejected by chance, not because of differences between experimental conditions, but simply by chance. So in a hypothetical example, in a trivial example, so remember we assign statistical significance when our alpha value gets below 0.05. So when we generally assign that, biological significance to effects where the probability of the null hypothesis being true is less than 5%. Okay, and so just by chance, if you had, say, 20 statistical tests, 1 in 20 by chance might be expected to be less than 0.05, just by chance. Okay, so when we conduct very large numbers of statistical tests, this becomes a particular problem, and we have to eventually recalculate the p-value to compensate for the multiple comparisons. That's effectively how we deal with this multiple testing problems, is we apply some method that will adjust our p-values that will make them less susceptible to this multiple comparisons problem. So, and the general outcome is that, most of these methods require that a test have a lower p-value in order to achieve the same level of statistical significance as a single test. So two methods that we can illustrate how this is done is one is the Bonferroni correction. This is not something that's generally applied in genomic research because it's too stringent. So what the Bonferroni's goal is to eliminate every single type 1 error. That is a case where we have falsely discovered or falsely rejected the null hypothesis when it is in fact correct. Okay, so that's really taking a hacksaw to the data, because when I've played with the Bonferroni correction, and you end up eliminating massive numbers of statistically significant or what appear as nominally significant cases, and so the Bonferroni correction is generally not applied simply because it creates a situation where you end up losing a lot of your statistical tests to non-significance. What's more generally adopted in RNA-seq analysis is the false discovery rate control, which is has greater power than Bonferroni and will commit some type 1 errors, but you're more likely to still retain enough genes to draw some conclusions about differential expression. And it does this by aiming to control the expected proportion of tests that falsely reject the null hypothesis. So finally, I'd just like to, wanted to make sure that you didn't walk out of this class without some introduction to the concept of RNA-seq quality control. And, this is a somewhat separate topic, and yet some of the same issues that we've been discussing in statistical analysis of RNA-seq data, are relevant here with spike-ins. So spike-ins are essentially a cocktail of molecular standards. These are known RNAs that have a known concentration and a known length. And so if you're working on a mouse or a human, you might use spike-in RNAs from a plant, or if you're working on a plant, you might use spike-in RNAs from a mouse. But essentially what these spike-ins is they're added to your RNA sequence, your RNA preparation, preferably during the library preparation step, and then they're just sort of carried through your entire RNA-seq sequencing protocol and analysis. And what they allow you to do is to have a set of known of molecules whose quantity is known, whose length is known, and you can evaluate then whether or not you're able to detect the certain say molecules that are found at very low abundance in the cocktail, and to see if you can quantify accurately spike-ins, particularly at the very low end of the range, and just where is your dynamic range. This type of spike-in analysis allows you to assess that. It allows you to assess where the linear parts of your quantification of RNAs is, and where you get so low that you're no longer in a linear range and therefore in a danger zone in terms of drawing conclusions about your real sample RNAs. So all of these issues here, quantification, sensitivity, coverage, linearity, can be somewhat addressed by including these spike-ins in your analysis. So I just want to point out that ideally these spike-ins are added early on in your experiment. They span the full length spectrum because, as I pointed out, length of molecules has presented some challenges to RNA-seq analysis, and they should consist of different classes of RNA, including mRNAs or long non-coding RNAs or other RNAs that might be in your transcript pool. And then finally, ultimately, when you do infer differences in gene expression from RNA-seq data, it's probably best practices to follow up at least with a small number of particularly important genes that you'd like to validate to make sure that your RNA-seq protocol and your statistical analysis hasn't misled you and that you've somehow misinferred the differential expression of some important subset of genes. And so the gold standard method for doing this is to conduct quantitative reverse transcriptase PCR, or qRT-PCR, sometimes just called qPCR. And this is the gold standard method that allows you to then, verify that in fact, your gene of interest is differentially expressed between conditions. And this is done, though, typically only on tens to hundreds of genes, hundreds being sort of the upper bound, more realistically tens of genes, simply because of the expense and the throughput considerations. And so this is not generally an approach that we're able to do on whole genome types of experiments. Okay, and with that, I'd like to thank you and for listening, and we'll see you next time.